Lovely weather out there. We are rapidly moving our way through translation, so happy to see that. Last time when I was finishing, I got started talking about the Shine-Dalgarno sequence. I want to come back to that and say some more about the Shine-Dalgarno sequence. So the Shine-Dalgarno sequence is what we could think of as, actually I won't even use those terms. What I like to think of the Shine-Dalgarno sequence is is an alignment. Okay, so it's an alignment to help tell the ribosome where translation starts. Now that's important because the first AUG that appears in a sequence is not always the right one for translation to start at. And so it's important that translations start at the proper AUG. You'll notice on this figure that there are occasionally GUGs that are used, and those are very rare, but the first base incorporated is still a methionine. All right? So don't obsess. In fact, I wish they wouldn't even put that on there because that's a very rare phenomenon where that occurs. But in any event, the point is that this conserved sequence is found roughly 10 base pairs or so away from the starting AUG. And as we will see, what this sequence does is it is complementary to one of the ribosomal RNAs, to a sequence within one of the ribosomal RNAs. And so that ribosomal RNA forms a base pair with this part of the messenger RNA. And in doing so, it places the AUG in the proper place in the ribosome for translation to get started. Okay? So that's a very important thing. By doing that, that means that the right AUG will, in fact, be the one where the translation of the message will start. Okay. This figure shows now those three sites that I described to you last time that I said were important during the process of translation. They are the E site, the P site, and the A site. And as I noted, the ribosome is moving down the messenger RNA. You don't really see the messenger RNA on here. You see it over here. The ribosome is moving from left to right so that new sequences are coming in first to the A site. Now, the A site, the P site, and the E site each are individual regions of the ribosome, and they each basically have 3 nucleotides that of the messenger RNA that will be exposed in each one. All right? So those 3 nucleotides of the messenger RNA are, of course, the codons that are being translated by the messenger RNA. And it's those 3 bases that will form the base pairing with the 3 bases of the anti-codon loop of the transfer RNA. Okay? So a transfer RNA, with the exception of the very first one. Of course, the very first one is F-Met, right? That's the one that starts off the whole process. F-Met starts in the P site. It's the only one that starts in the P site. All of the other transfer RNAs, when they come in, they will first come into the A site. Okay? And we will see that this ribosome will go shuffling along, moving down the messenger RNA and shifting the positions of those transfer RNAs as it's moving along. The E site really serves one and only one function for our purposes, and that is that it is the exit site. That's what the E stands for. And so it's just basically a place to unload the transfer RNA. Now notice that the transfer RNAs that are here, you'll see this actually when I show you the mechanism later, the transfer RNA that's in the P site will have the growing polypeptide chain attached to the tRNA. The tRNA that's in the A site will have the next amino acid attached to it that will be added to the chain. And the one that's in the E site will have no amino acid because it's already dumped its load and it's on its way out of the ribosome. Okay? So that's the sort of playing field on which we'll be describing the process of translation in this lecture. Okay. This schematically shows the very same thing that I just told you. And so you can see they've color-coded them here. And you can see, again, this transfer RNA that is leaving through the E site. You see the transfer RNA that has the growing polypeptide chain. And this could be many amino acids long. It is emerging from that tunnel in the 50S ribosome that I described to you last time. And of course, the transfer RNA that's in the A site has the new amino acid that's coming in. And so what will happen during the process of translation is a peptide bond will be formed between the green guy and the purple guy. That will result in the growing polypeptide chain being shifted from this green transfer RNA onto the purple transfer RNA. All right? And then everything will sort of shuffle down, as we shall see. Okay. So what I want to do is spend some time going through, going through this process, and I am missing, here's what I want. Going through the process of translation initiation, elongation, and termination. And we're going to do this in some detail. Okay? So this shows the sort of steps in the initiation process. It turns out that for initiation, the ribosome has to be assembled onto the messenger RNA. The ribosome has to be assembled onto the messenger RNA. So this starts with the small ribosomal subunit, the 30S subunit in prokaryotes. Of course, this would be the 40S subunit in eukaryotes. And there are 3 proteins called initiation factors that help to make this process happen. Okay? So the first initiation factor is IF1. And in combination with IF3, IF1 and IF3, well, actually, I should back up. IF1 and IF3 help IF2, which is the last initiation factor, to bring in the messenger RNA and the Met-tRNA. So these 3 initiation factors, I won't distinguish the functions of 1 and 3, but I will tell you a little bit more about 2 in just a second. IF2 has a very important function. All right, remember that the transfer RNA has a bond to its amino acid that is very unstable in water. And I described to you last time a protein I called EF-Tu, EF-Tu. All right? And its function was to protect that bond so that water didn't get to it. The only exception to that is the Met-tRNA. Instead of using EF-Tu, it uses IF2. All right? So IF2 is functioning like EF-Tu, and it makes it very easy to remember. It's a 2 and a 2. Right? It is covering that bond so that water isn't exposed to it. All right? And it's helping to assemble this overall complex that is getting started. All right? Now, IF2 is a protein we call a G protein. All right? A G protein. We talked about G proteins last term in a eukaryotic sense and in a very different kind of a sense. Okay? Those were very important proteins in the process of signaling. All right? But the term G protein is very broad. It simply means a protein that binds guanine nucleotides and uses their energy. So that's what this guy is doing. IF2 is a G protein. You can see it's bound to GTP. It's also bound to the Met-tRNA. So after this guy all gets together right here, okay, what we see is a complex that looks something like this. This forms what we call the 30S initiation complex. And the 30S initiation complex at that point is ready to get the 50S subunit. Addition of the 50S subunit requires the loss of IF1 and IF3. Okay? And as it comes in, IF2 also leaves. IF2 leaves as the 50S subunit comes in. So IF1 and IF3 leave, then the 50S subunit comes in, and as it comes in, IF2 leaves. As IF2 leaves, it cleaves GTP to produce GDP. Okay? So the energy of that GTP is used. It's used partly in helping to assemble this complex between the 30S and the 50, the overall ribosome, the 30S, 50S complex. At this point, we have finished the process of initiation. What we have is the full intact 70S ribosome. We have the messenger RNA. We also have, and I didn't mention it up here, but when we brought in that messenger RNA, we also aligned the Shine-Dalgarno sequence to the 16S ribosomal RNA. That's the 16S ribosomal RNA. And that 16S ribosomal RNA is contained in the small subunit. So there's a base pairing that's going on over here just to the left of the AUG. That's where the ribosomal RNA, the 16S, is bound to the Shine-Dalgarno sequence of the messenger RNA. So that, as a result of that, the AUG is placed in the proper site. And by the way, that proper site is the P site. That's the only transfer RNA that comes into the P site. At the end of this process, as I said, we have the 50S complex, we have the messenger RNA, we have the AUG in the P site, and it is base paired to the F-Met transfer RNA. At this point, we are ready now to proceed with elongation. That's where we're actually going to make the protein. So right now, we're all primed, we're all ready to make the protein. Okay? Questions? I see some yawns. Man, it's a long day, isn't it? Good sleeping weather, right? Yes, question back there. Why does it say 70S? Well, as I said last time, they're not additive. So 30S and 50S are not additive. They are relative sizes of things, 50S being larger than a 30S, but you can't add the two. All right? So 70S is what you get when you put those two together. Okay? Same is true of eukaryotic ribosomes, which are 40 and 60, and they give you an 80S. Okay? Yes?
Could you just repeat like an overview of what the initiation factors do?
Okay, an overview of what the initiation factors do. For our purposes, they all help to set up the complex between the small subunit and the messenger RNA. That's one thing that they all do. The IF2 specifically carries the F-Met-tRNA and places it on the AUG start codon. There's two things. Okay? Then they basically exit. 1 and 3 leave as a precursor to the large subunit coming, and as the large subunit comes in, it displaces IF2, and IF2 cleaves its GTP and allows the overall complex to form. Does that help? Okay. Okay. Other questions? All right, so we have started the process of translation. All right, now, this figure now shows the process of elongation. All right? So we've got the very first transfer RNA with the very first amino acid in the P site. You'll notice that's depicted up at the very top. That's the very last step of the previous process, which was initiation. And you'll notice that the A site is wide open. There's nothing in the A site. Okay? That means that it's open for the next transfer RNA to come in. And the next transfer RNA comes in, and it's bound to the protein EF-Tu. EF-Tu. By the way, IF stands for initiation factor, so there's 3 IF proteins. EF stands for elongation factor. And it turns out there are 3 elongation factors as well. Okay? Now, they're not numbered. The EFs are not numbered. But, and in fact, we only talk about 2 of them right here. I'll talk about 2 of them. All right? So EF-Tu is one of them. EF-Tu has 2 important functions. First, it is covering that bond that is very unstable between the transfer RNA and the amino acid. Okay? EF-Tu is also a G protein. It carries GTP with it, and that GTP plays an important role, as we shall see. Now, what EF-Tu does is it brings that transfer RNA to the A site of the ribosome. But EF-Tu, just like everything else going on here, doesn't have a brain. All right? So it will bring in a transfer RNA, and then it asks the question, did the transfer RNA I put in the A site make the proper base pairs with the codon that's there or not? If the proper base pair formed, then everything's fine. All right? And GTP gets cleaved and EF-Tu leaves. Now, it's leaving it within the confines of the ribosome, so still water hasn't had a chance to come in and attack that bond. If, on the other hand, that codon that is in the A site does not form proper base pair with the tRNA, it means that EF-Tu, this isn't the right one we want in here. What EF-Tu will do is it will take away the tRNA. So EF-Tu is the quality control to make sure that the right transfer RNA is in the A site. Okay? Only if the right one is present will GTP be cleaved and EF-Tu will leave. If it's not the right one, then EF-Tu says, sorry, I jumped in too early, takes this transfer RNA away, and leaves the site open for another one to come in. Okay, well, assuming we've got the right one in there, we have the situation that you see in the second panel there. That is, that the P site has the transfer RNA, in this case with just F-met, and this guy has the next amino acid specified by that codon. We are ready at this point, then, for peptide bond formation. That's going to join the two amino acids. Now, look what happens. As I said before, the one that's in the middle here, the blue guy, is going to be linked to the green guy. All right? So look at this. Look at what's happened. We see a shift that's happened, right? So that the top part of this green guy now reaches over into this tunnel and makes that peptide, and the peptide bond is made. That peptide bond is catalyzed, as I said last time, not by a protein. It's catalyzed by the 23S ribosomal RNA. The 23S ribosomal RNA catalyzes the formation of that peptide bond between these two guys, and we're left with this complex looking like it is here. Well, we can see this guy has already lost its amino acid. It's going to be on its way out. It's going to move over to the P site in a second. And the way that it moves over to the P site is as a result of the ribosome shifting. The ribosome actually shifts 3 bases at a time, moving from left to right. So it's going to shift, and when it shifts, we're going to see now the blue site, which was over here in this part, is now one more over. It's now fully in the P site. Here it's halfway in the P site, in the E site. Here it's fully in the E site. This guy was halfway in the P site and halfway in the A site. Now it's fully in the P site. The movement of that ribosome is catalyzed by the second G protein. It's called EF-G. And you'll notice again that GTP is the energy that's used. GTP is the only energy that's used in translation. ATP is not used in translation. GTP is the energy source of translation. Okay? Well, we've got everything ready now. This guy's in the E site. The blue guy's in the E site. It has no amino acid. It's ready to exit, and it exits. We are then left with the situation we have down here at the bottom. We have a single transfer RNA now with 2 amino acids on it in the P site. That's equivalent to what we had right up here where we started. So we go from here up to here, well, here to here, all right? And a new transfer RNA will come in. So this process is repeated on and on and on until a stop codon appears in the A site. This will continue on and on and on until a stop codon appears in the A site. When a stop codon appears in the A site, we are ready for the process of termination. Okay? Now, before I go to that, I'll take any questions you might have. You have a question?
Why is GTP used instead of ATP?
People asked me that earlier. Why is GTP used instead of ATP? Because it works.
And how is it regenerated? How it would normally be regenerated?
So how is GTP regenerated? Okay, so that'd be a good exam question because you actually know the answer to that question. How is GTP generated in the cell? NDPK. Okay. So how does NDPK make GTP? It's using the energy of another triphosphate to put a third triphosphate onto GTP. So NDPK, remember, can take any nucleotide and use its triphosphate to put it onto another one that only has two. So that's how GTP is made. Okay? Yes? How did I say proofreading occurs? There's no proofreading that's occurring. Okay, so, well, I'm sorry, maybe you're asking about EF-Tu, how it's making sure the right base pairing is happening? Okay, well, let me just clarify, since it's sort of a semantic argument. I don't use the term proofreading associated with what's happening here, okay? This is a base pairing phenomenon. Proofreading I exclusively used when I described how the amino acyl tRNA synthetase was putting the right amino acid onto the right tRNA. All right? I'll answer your question here in a second, but I want to make sure we're clear on that. So we talk about proofreading in this overall process related to translation. Proofreading relates to the putting of the proper amino acid onto the proper tRNA. And that's a function of the amino acyl tRNA synthetase. This process, which you're asking about here, this process is quality controlled, might be a good way to say it. It's quality controlled as a result of the action of EF-Tu. EF-Tu, remember, is bringing in the appropriate transfer RNA that has the right amino acid attached to it, and it is placing it into the A site. And then, when it places it in the A site, it's actually able to check, did the proper base pairs form here or not? So if this thing is properly base paired, then it's got the right tRNA, and if it's not properly base paired, it doesn't. So if it doesn't have the right tRNA in there, EF-Tu takes it away. Does that make sense? Okay. Yes? Each colored block in the messenger RNA refers to 3 nucleotides which are a codon. That's correct. Yes?
Is GTP used to halfway put the new tRNA into the P site as well?
Is GTP used to put this into here? Yes. No, GTP is simply used to do what's called translocate, which is to move the ribosome further down. So you can see that this guy is leaning over. I mean, actually, this has to lean over a little bit during that peptide. It's called a peptidyl transferase, that is the making of the peptide bond. This has to move over a little bit in order for this bond formation to occur. But that's not requiring GTP, no. How does it shift it over? Good question. What happens inside of enzymes? How does enzymatic, what makes enzymes more efficient than chemical catalysts? Flexibility, right? So we've got not only flexible proteins, but we've got 50 of them here, all right? So we can imagine this guy is literally a breathing thing, and this breathing allows for movement and allows for motion, and that's how it gets moved over, okay? Yes? How is the messenger RNA released? I'm coming to that in termination, okay? Yes?
So what is the source of the energy for the breathing? Like moving around the
What's the source of the energy for the breathing? What was the source of the energy for the enzyme being flexible?
shape
the Brownian motion of all the atoms that's occurring inside there. So that's ultimately what it takes no energy for that to happen. Okay? Okay. All right. So we have now finished, or we're finishing the process of elongation. There's translation up close and personal. And you can see that EFG comes in and it actually invades the A site. Part of it looks like a transfer RNA. That's how it fits into the A site. And in fitting into the A site, it's actually butting its way into there and kicking everything over. That's how translation is happening. So though I said the ribosome moves, the movement is made possible by the action of EFG. EFG is making that happen, and it's making that happen by sort of squeezing its way into that A site. Okay. EF-Tu. Why does that say EF-Tu? That's mislabeled. Yeah, okay. I'm missing a link for EF-Tu. All right. This now shows the process of translation. Okay? So, termination of translation. So, we have reached a point where a stop codon has appeared in the A site. In this case, we have a UAA. Okay? This UAA that is here doesn't have any complement to any transfer RNA that's there. So it sort of sits there for a bit. Remember, we're talking relatively short periods of time because translation is moving at 17 amino acids a second. This thing sits here for a couple seconds. That's a bit. It's not sitting here very long. But it sits there, and this gives the opportunity for a protein called a release factor to come in. A release factor can come in. All right? There are release factors specific for each stop codon. Okay? And they can form base pairs kind of like what you see right here. Now, the release factor's primary function is to release the peptide from the last transfer RNA that's in the P site. It brings with it a molecule to facilitate that process. What molecule do you suppose it brings? Water. Water. It's carrying a water so that it says, okay, now is the time we can break this bond, because remember that's a very unstable bond that's there. It carries water, and when water hits this guy, what does it do? It cleaves it, and the polypeptide is released. Now, if the polypeptide hasn't already started folding, which it usually will have, if it hasn't already started folding, it will definitely start folding at this time. The process of folding occurs fairly rapidly. The number range that people give for the process of folding to occur is between 0.1 and 1,000 seconds. Okay? 1,000 seconds is a fair amount of time in a cell. But nonetheless, 1,000 seconds really only corresponds to about 15 minutes. All right? And we think about the complexity of the process that's going on. That's not a giant amount of time. Most proteins will fold very quickly. Okay. Well, at this point, we have a ribosome now that has no polypeptide chain attached here. And that, complex now will interact with another protein that comes in, which I won't give you here, but another protein comes in and basically disassembles everything. It releases the tRNAs, it releases the 50S ribosome, and it releases the messenger RNA. Okay? And that's the function of that last protein that comes in. Okay? At this point, we have a messenger RNA that's sitting there with nothing on it. Now, one thing I haven't told you that I need to tell you, and it's a very important consideration, is that when we look at prokaryotes, they have interesting and unusual messenger RNAs compared to eukaryotes. Eukaryotes have a pretty specific rule. One protein for one messenger RNA. Prokaryotes don't have that rule. Prokaryotes commonly will have more than one coding for a protein on a given messenger RNA. Some have as many as 10 on a given messenger RNA. Okay? This organization of multiple coding regions on a single messenger RNA is referred to as an operon. Okay? And I should back up and say I put this on a messenger RNA, and technically an operon is not a messenger RNA. Technically an operon is the part of DNA that gave rise to that messenger RNA. Okay? So the operon was the section of DNA that contained all of those genes. Now, this becomes important, and now we begin to see why there are Shine-Dalgarno sequences. Because we don't have to worry about translating just the first protein. There may be other proteins further down for this messenger RNA to code for. So this means that this ribosome assembly that we talked about here may have to reassemble further down the messenger RNA because it says, hey, look, here's another Shine-Dalgarno sequence. Let's start the process again. So at any given time, a given messenger RNA might have several different proteins being made on it at the same time. Yes?
So do messenger RNAs in eukaryotes have one Shine-Dalgarno sequence or not?
So in eukaryotes, there are no Shine-Dalgarno sequences. And I'm just getting ready to talk about that. But the eukaryotes do not use the Shine-Dalgarno sequence. And there's a good reason why, and I'll show you that in a minute. Okay? But eukaryotes do not use the Shine-Dalgarno sequence. Other questions? Okay, so we have now finished prokaryotic translation. So I want to spend a few minutes talking about eukaryotic translation, and kind of like I talked about with respect to DNA replication, the overall process is very similar. I'm only going to show you the differences, and I'm not going to expect you to know the names of the eukaryotic factors, because, again, it's just a purpose of memorization. I don't think if you know the prokaryotic ones, you have a pretty good idea about what the names of the eukaryotic ones are anyway. There are more factors. There are more factors in eukaryotes than there are in prokaryotes. All right? But again, the overall process is very, very similar. When we look at the eukaryotic process occurring, as I said, I want to show you what some of the differences are from prokaryotes. First of all, in eukaryotes, the very first amino acid that's put in is not F-Met. It's methionine. Now, you may remember I told you that methionine had that free alpha amino group. It's the only one that has a free alpha amino group after it's been starting the process for making a protein. And the formyl group in the case of prokaryotes was put onto there to cover that alpha amino group. Eukaryotes don't need that because their ribosomal structure covers that part, the alpha amino group. It doesn't allow it to be able to react undesirably. So F-Met is not needed in eukaryotes. All right? That's one difference that starts. Okay? The second difference is that if you remember, messenger RNAs in eukaryotes have two distinctive features. They have a 5' cap, and they have a 3' poly-A tail. Prokaryotes do not have that. Okay? The cap is recognized by the initiation machinery as, all right, this is the right end of the molecule. And we can see that the small ribosomal subunit is getting positioned down there. Okay? Well, I said there's no Shine-Dalgarno sequence in a eukaryotic messenger RNA. So how does the ribosome know where to start? Okay? Well, it turns out that there are some sequences that help the ribosome to understand where to start. They're called Kozak sequences, K-O-Z-A-K. But even those aren't very specific. So how does the ribosome know where to start? Well, usually in eukaryotes, the first AUG is where translation starts. So all this thing really has to do is slide until it finds the first AUG. Okay? Now, you see some involvement here in the initiation process of setting this thing up using ATP, okay? And that can happen in eukaryotes, all right? But again, primarily GTP is used for the process of translation. We see some scattered ATP that's here, all right? There's the AUG. We see the sliding down. We're sliding down to get here, and finally we have this guy positioned in the right place. We have what will be the P site, which is right here. Okay? We have the transfer RNA base paired. There's the initiator methionine. And then the initiation factors leave just as they did in the prokaryotes, and the larger ribosomal subunit comes in. In the case of the eukaryotes, the small subunit is 40S, the large subunit is 60S, and this overall complex is 80S. We're just exactly at the same place we were at the end of prokaryotic initiation. Eukaryotic elongation occurs very, very similarly to prokaryotic prokaryotic elongation. It uses factors like EF-Tu and EF-G for both bringing in the transfer RNAs and translocating that ribosome along the messenger RNA. So I don't even bother to show you that process here. Similarly, translation proceeds until a stop codon appears in the A site. And when that happens, release factors come in just like they did in prokaryotic systems, and exactly the same thing happens. It releases everything, and everything falls apart. Okay? So very, very similar overall to the process that's going on inside of prokaryotic cells. Now, there's one other interesting difference in eukaryotic cells that I want to point out to you. And that interesting difference actually relates to the fact that there's a cap and a poly-A tail. This shows that the poly-A tail, which is over here, binds to some proteins, and those proteins interact with initiation factors. That stands for eukaryotic initiation factors. No, you don't need the numbers or anything like that. All right? These initiation factors recognize the cap. These factors down here recognize the tail. And what they do is they form a circle. Why do they form a circle? Why does the messenger RNA translate it in a circle? Well, it's not completely clear, but people tend to think that the reason that the circle forms is once the ribosome goes along down over here and it reaches a stop codon and everything falls apart, these things falling apart are relatively close to the start. This is kind of exaggerated in terms of size. It's relatively close to the start. So the components don't have to go very far. It ensures that translation will proceed more efficiently. Okay? It ensures that translation will proceed more efficiently. So that's one thinking about why there's a circular messenger or circularization of this whole complex to make translation occur more efficiently. Okay. Questions about that or about eukaryotes? Yes? I'm not sure I understand your question. Say it again. I'm still not hearing you. Yeah. Yes. It goes in tunnels. Uh-huh. How can the releasing factor release the peptide? Okay, because it's not going inside the tunnel. It's going down to the A site and the P site. So the releasing factor is coming into A site, and that's immediately adjacent to the P site. So if it releases a water directly into the P site, that is going to hit that bond between the transfer RNA and the polypeptide chain to release it. So the tunnel has nothing to do with that. Okay? Okay. Other questions? Yes? Okay, so clarify the difference between prokaryotes and eukaryotes with respect to how they use the cap and the tail? Okay. Well, the primary difference is the fact that prokaryotes don't have a cap and a tail. So prokaryotes are always going to be translating a linear messenger RNA. So let's imagine that this guy is, very long. So when the translation of a prokaryotic ribosome gets all the way to the end and it releases its cargo, that is, its large and small subunit, these guys are a long ways away from where the start site was over here. In the case of eukaryotes, it's circular. So if it starts over here and it gets over here and it releases them, those units only have to travel a short distance to where the start site was. Does that make sense? No. Well, okay. So his question is that the reason that there is a 5' cap and a 3' poly A? Well, that's not completely true because if you recall, I said that the 5' cap protected against exonucleases and the 3' poly A tail also gave added life to the messenger RNAs. So there's more than one function associated with both of those. Okay? Yes?
So in the prokaryotic translations, the stop codon on the mRNA doesn't necessarily release the mRNA?
So the question is, does the stop codon release the messenger RNA? And the stop codon itself does not release the messenger RNA. It takes the release factors in order for that to happen. Yeah, right. If there's no release factor that comes, it'll just sit there. Sitting there waiting for something to happen. So when it's translating several polypeptides, it's still going to reassemble that ribosome further down. Yeah. But again, if we think about this, they're not too far apart, right? So it doesn't have to travel very far to the next one, right? Yep. Okay. Other questions? Good. Getting through this nicely. All right. Translation is a very, very good target for antibiotics. In fact, probably the process of translation is targeted by more antibiotics than any other process in the cell. Okay? Now, you say, well, how can we do that? Well, the way we can do that is that there are enough structural differences between the ribosomes of prokaryotes and the ribosomes of eukaryotes that we can specifically target things there. Some translational inhibitors will kill both types of cells because it's inhibiting a very general process. Some target specifically things that are found only in prokaryotic cells. So streptomycin is a really good example. Streptomycin interferes with the binding of the 4-methionine tRNA to the ribosome. We don't have a 4-methionine, right? So if you target only 4-methionine binding to a ribosome, you're not going to affect us, but you're going to affect every cell that uses 4-methionine, and that's going to include all the bacteria. Okay? Very, very simple thing to do. There are numerous antibiotics, as I said, that target different specific things. So, for example, tetracycline binds to the small subunit and inhibits the binding of the aminoacyl tRNAs. The structure of that unit is distinct enough from ours that tetracycline doesn't affect our translation, but it does affect translation in prokaryotes. Okay? Chloramphenicol inhibits the peptidyl transferase, that's the formation of the peptide bond, okay, within the large subunit. And that process is, again, distinct enough from ours that only prokaryotes are affected by that. Some things only affect eukaryotes. Cyclohexamide. If you take cyclohexamide, you're not going to be in very good shape because it's going to knock out translocation, and translocation is that movement of the ribosome. You're not going to translate proteins if you do that. Okay? If you look at puromycin, it affects both of us because it causes chains to terminate prematurely, and there aren't distinct differences in those processes between prokaryotes and eukaryotes. Okay. So those antibiotics are very effective. Streptomycin looks like this guy right here. Don't have to worry about structure. Puromycin is one I like to show because it resembles an aminoacyl tRNA. It resembles it close enough that it can actually get into the A site, and the amino acid, which is right here, can be attached to that growing polypeptide chain, but when it does, this is not a tRNA, everything flies apart. So consequently, elongation stops at that point. You stop translation immediately as soon as this guy gets incorporated into a growing polypeptide chain. And let's see. Ricin is one that's in the news. It's a bioterrorism weapon. It cleaves adenine from the 28S ribosomal RNA. 28 ribosomal sRNA is in eukaryotic cells. It's the equivalent of the 23S ribosomal RNA in prokaryotic cells. And it prevents the binding of elongation factors. So if you prevent binding of elongation factors, you're going to stop the process of elongation. Okay, the last thing I want to talk about very briefly is protein secretion. Protein secretion is something that's going on in our cells all the time. And protein secretion is directed through organelles. So it's going through the endoplasmic reticulum, and so we need to understand how it is that translation is occurring. If we look at the endoplasmic reticulum, we can see what's called rough ER. That rough ER has all these little bumps along the outside, and what those are ribosomes that are translating protein. Ribosomes are translating protein, and what they're doing is they're injecting the protein that they're making into the endoplasmic reticulum. They're injecting it into the endoplasmic reticulum, and we'll see right now how that happens. That happens as a result of action that arises from what are called signal sequences. At the amino terminus of a protein, there are sequences that may not look very conserved, but these are basically non-polar amino acids that are preceded, for the most part, there's only one exception here, that are preceded by basic amino acids like lysine, like arginine. This provides a signal to the cell that this protein is for targeting for some place. It needs to go to the endoplasmic reticulum. What those sequences do is provide a binding site for something called the signal recognition particle, or SRP. Okay? Don't worry about this complex here. We won't talk about that. But the SRP helps this overall process to happen. Here's a messenger RNA that's being translated. Out of its 50S large subunit emerges the new protein being made, and there's that amino terminus with these amino acids within them. The signal recognition particle says, this guy is going to the endoplasmic reticulum. So it grabs them and drags it down to an SRP receptor in the endoplasmic reticulum's membrane. Then it's pretty straightforward. This redirects the polypeptide chain through this thing called a translocon, and we see the polypeptide being squirted into the endoplasmic reticulum as translation proceeds. Because this had that sequence, the SRP recognized it, and the SRP recognized this, and the protein got dumped into the endoplasmic reticulum. Okay, I'll repeat this at the beginning of next time, but I do have a song I'm very fond of that I want to play for you. It's called Translation. Surprise. And it goes as follows. I can't sing it. Translation to form peptide bonds in translation. The ribosomal cleft must bind to an EF-Tu. Translation. He binds the F-Met's cargo. 16.